All right, protein synthesis from the beginning, from soup to nuts. You ever hear that expression?
Nope.
Probably not, and that's okay. I'm not going to go through it. Because if I do, Bulkley's got to come up with something if I use the word nuts. So I'm not even going to bother. You notice I'm giving him all kinds of, I'm going to throw out all kinds of softballs here, and so he can hit them out of the park. Wait, baseball. Baseball! Cheaters! He's got to say something because he's getting points off every single time. All these softballs are going up there. All right, DNA. What is DNA? Now, I hope you all remember what Lisa stands for. What does it stand for?
Deoxyribonucleic acid.
Yes. And it is the ultimate source of information in the cell. It's the ultimate source of information, and we basically read it like you would read a sentence. It tells the cell what to make, what protein to make. Okay? And different cells make different proteins. Like I said, I think before, a blood cell is going to make hemoglobin. A pancreas cell is going to make insulin. All right? And those things have a job to do in the cell. The hemoglobin carries oxygen. It does its job. The insulin, what does that do? Lowers your blood sugar, right? All right. So all these things in your body, everything in your body is basically run by proteins. Why is your hair brown? Why are your eyes blue? All has to do with proteins. All right. It's the ultimate source of information for the cell, and it's used to produce protein molecules, like I just said, which basically are responsible for every function in your body, everything you do. Your movement, your seeing, your hearing, your smelling, all has to do with specific proteins. Now you can see on there while you're copying that down, it's a lot to copy, is the shape of the DNA molecule. You've all heard of that. What's it called? What's that?
Helix.
The double helix. It's like a ladder that's been twisted. Okay? And that ladder can come apart in the middle. And that's what we're going to talk about, how it does that, why it does that, to be able to be read and actually make proteins. So first, let's talk a little bit about the structure. And I just added a couple of pictures on here because I realized how I'm using. I originally did this 3 years ago when I was teaching bio. And then I also kind of condensed it when I was doing your Bio Keystone reviews. All right? But I realized that some of these pictures are pretty basic, pretty simplistic. So I found a couple more pictures. I'm going to try to expand on a little bit from what I had back then. So when you see this picture here, that's such a terrible picture. I'm going to show you what these things really look like in a minute. But I know that's the one that's on your paper. The phosphate group is a blue circle. And the deoxyribose, there's a reason, by the way, that it is a pentagram here, and pentagon, I mean here, and that one looks like that a little bit. But that's not really obviously what they look like. And I want to show you some other pictures or some other things about it in a minute. But they are the actual nucleic acids. Okay, that's what these guys are, nucleic acids. You put the phosphate group with a deoxyribose sugar and some one of the purines or pyrimidines, and you have a nucleic acid. That's what makes up DNA. DNA is a huge molecule. It's probably one of the biggest molecules that we know of. Biggest, largest macromolecules in biology, period. Okay? Because if you think about it, the actual molecule itself is all, if you were to stretch it out, okay, because it gets coiled up into a chromosome, then I could actually see under just a microscope. We did a lab when I was in college, and you may have done too. I don't know. You guys ever work with fruit flies in bio? No, well, I know they have them over there. I've seen them order them, and I don't know if it's in bio 2 or AP bio. But you can actually get the salivary gland, I believe it's salivary gland, chromosomes from there, from cells in there that are huge, and you can actually see them under a microscope. Now think about that. That's how long that coil of DNA must be packed and dense and coiled on top of itself in order for you to see it with just a simple microscope. Okay? It's a very long strand of nucleic acids, and they are composed of 3 parts. Okay? Those 3 parts are, as you can see on here, I'm going to show you a better picture in a second, the phosphate group, the sugar group, and the nitrogen space. Anybody taking bio? I'll tell you what, keep these notes if you're taking bio 2 or AP bio next year. I can't imagine you're going to get any more in-depth treatment than you're going to get from this.
Who teaches them?
I'm not sure. I think it could be Mrs. Rothery or Mrs. Saragotis. I mean, Marinakis. That's her other name. Okay, so if you do take it, don't lose these notes. I think they'll come in handy. Those are the parts. How do they work? What do they do? How do they make up the actual ladder? Let's take a look. Well, first of all, let me show you better pictures of these. The phosphoric acid looks like this. All right, better. The purine or pyrimidine base, that's actually not as good a picture. You're going to see better ones than that in a second. This is not on your thing. And notice why the sugar was drawn as a pentagon? Because it actually, in the body, even though if you stretch it out straight, it can be drawn as a straight chain. In the body, it actually coils into a ring structure in a water situation. All right, and when you put those three things together, you get a nucleotide, which are bound together like this. Now, that nucleotide makes up, when you link them together, makes up the ladder. Now, this is the page you should be on now. Okay.
Yeah.
The only difference, this is the key, the only difference between each nucleotide we just saw is this guy right here, the base that's over here. Everybody's got the same sugar, deoxyribose. By the way, it's called deoxyribose as opposed to ribose. Where do we use ribose in? Ribonucleic acid would be what? RNA. We're going to talk about that. Deoxyribose just has one less oxygen. That's all. It has everybody's phosphate's the same. It's this that's different, the nitrogenous base. What are the possibilities? You've all seen this before too. Adenine, thymine, guanine, and cytosine, right? And they have to pair up that way. Okay? That's the only difference between these guys. All right, the purines, which are these guys over here, adenine and guanine, they have a double ring. Notice they've got like a hex ring and then a pent ring, which makes them longer than the thymine. Now, if you think about the ladder, how it would look, all right, they have to match up a certain way. The pyrimidines have a single ring, and they have to match up a certain way, otherwise the ladder wouldn't look right. It wouldn't fit together correctly. I'll draw that for you in a second. Copy that down. A lot of times when you see models of these guys, you actually see that the purines are written as a longer rung of the ladder, and the pyrimidines are written as a shorter one, because they actually are. I'll show you that in a second.
Don't one of them in one of the different transcriptions?
When it gets to RNA, we're going to replace one of those. Yep, we'll talk about that too. Give me a second here. All right, I'll come back to this in a second. But look, here's what I'm saying. See how long this guy is versus how short this guy is. Here's the problem. If I've got my ladder, all right, one rung, let's use red for the purines, all right, they're longer rungs, all right, and the other one, we'll use pyrimidines, are shorter. Okay, so on the other side of that, if I have a blue with a blue, they're not going to hit, right? Or if I have a red with a red, it's going to be bulged out. So you have to have the blue matched up with the red, and then a red matched up with the blue, just for distance-wise to keep that ladder the same. You see what I'm saying? Yeah. And that's basically why they match up only as. And by the way, here's another picture. Again, I'm going to probably work these in over the course of, it's not on your paper, over the course of my change of notes. It's the first year again I'm doing this. A better picture perhaps of these guys, and maybe a better picture of the deoxyribose and the ribose sugar. Notice the only difference is one oxygen missing. But look at these guys have to be matched up as AT or GC or it won't work. If you don't do it that way, it won't work. Okay, so the backbone of the ladder, I guess I'm talking about amount of order here. The backbone of the ladder are these bonds here, the ones that are going along here. These bonds are just covalent bonds, and they should not break. You don't want them to break. Okay, we want them to stay where they are. These bonds in the middle, we're going to talk about, they're just hydrogen bonds, and they break relatively easily. And that's good, because that's how it gets copied. That's how the DNA gets copied.
So the bonds in the middle are easy?
Yes. These hydrogen bonds are very easy to break, and you notice they have to be a double ring with a single ring, a double ring with a single ring, whichever one. It doesn't mean the double ring has to be on one side or the other, and depending on how you read it, it's going to be T, G, A, C. Anybody could be on this side. Like I could have T, T, T. I could have 3 T's in a row. I got 3 G's in a row, 3 A's in a row. It doesn't matter, but opposite it has to be. If it's a T, it has to be an A. If it's a G, it has to be a C. Okay? So the rungs of the ladder are linked by these weak hydrogen bonds. And that's good because, you know, that's how we're going to end up copying this and transferring that information from the nucleus of the cell in the DNA to the actual proteins that the cell's going to make and do its job. And again, these bases only pair up this way, A with T and G with C. All right, on the last page, I'm not going to end up starting the transcription. I'm going to show you the video on it. We'll probably start on Monday with that same basic video on it in a little bit. But before I do that, a little bit of history here. Okay, you guys should know who those two guys are. Who are they? Watson and Crick. You've heard of them. There's probably about 3 or 4 biologists that you really should know. These guys. What are some other biologists you probably should know for what they did?
Discovered cells.
No, Mendeleev was the periodic table. You're thinking of Mendel. Remember for genetics, Mendel? They worked the pea plants. And Darwin, of course. And who else? You said about cells. That could be a that's actually Hooke or Leeuwenhoek. Right. Yeah. So there were those guys that first discovered cells for soft cells and cork cells. I think that was Hooke. What's that? Well, these guys, they're the ones, Watson and Crick are the ones that built, well, they actually built upon a lot of other people's work. And they came up with the 3-dimensional model we kind of know it looks like today, the double helix. Not a big fan of Watson and Crick. They're kind of like arrogant people. They really didn't give a lot of credit to other people who did do a lot of the work, number one. And number two is they're kind of like nutcases, too, one of them in particular. I wouldn't call him a nutcase. I'm sure he's much smarter than I am. However, he's one of those people who thinks that aliens seeded the Earth with DNA. That's his thoughts on where life started.
Scientology.
Yeah, that could be right. It could very well be one of the things, one of the bases of their tenets of their faith. And it's funny. I called it a faith by accident, but it really is. One of the things about DNA is it's so complicated and it's so difficult to envision this being created from scratch in a slow evolutionary process that nobody has any idea where the first cell came from, the first self-replicating DNA-containing organism. Once you have that, you know, they can go through a whole bunch of transitions to try to come up with mutations and natural selection and all that stuff to explain a lot. But where the first cell came from and where DNA comes from, no one, and I mean no one, has any idea. They have no idea. And that's the thing about a scientist. They're just like other people. You know, they basically want to explain everything, and they can't. And so they kind of, you know, they make compromises. And sometimes, like one of the things I hate is when people say that they know with certainty. You've heard me say this 100 times. Everything about from global warming to all those other things we've seen in the videos. Same thing's true here. No, let me give you a hint here. No one knows where the first cell came from. No one knows where DNA came from. I've got a degree, but I'm not a molecular biologist. But I know that much. Okay, I know no one knows. And anyone who says we know where it comes from is lying. All right, and they can have theories about it, but nobody knows. It's just and you can have a lot of theories about once DNA is there, what happens. But how you get that first cell, that first self-replicating organism, nobody really knows. And so guys like this, they punt. They make fun of religious people for punting, and they do the same thing. Well, we don't really know where it came from, so it must have been aliens who did it and dropped it on the earth, and then from then on. Well, that's just punting. That doesn't explain to me where the aliens got the DNA from.
I guess
So
when we're dead, we'll find out.
these guys, exactly. These guys are not my favorite people, but anyway, you have to give them credit. They did come up with the double helix strategy. All right.